Our first session is the first of 4 research introductions that follow the Hub's 4 priority areas, starting with 3 talented researchers with projects related to animal health and welfare. And remember, if you have questions, you have a chat function on your media player. So go ahead and put those questions into your media player and we will route them to the speakers at the end of the session. Also, as a reminder for folks who may have just joined us, we do have a student innovation competition going on right now simultaneously. Make sure to use the link on your info tab in your media player, and you can check out those videos and vote online for your favorite. So without further ado, we're going to kick things off with Mark Levenstein from UW-Platteville. Welcome to Mark.
Good morning, everyone. On behalf of myself and my research partner, Dr. Gokul Gopalakrishnan here at UW-Platteville, I'd like to thank all of those who conceived of, implemented, and launched the Dairy Innovation Hub. I think it's a great idea that resonates so well with Wisconsin's central identity. And I'd also like to thank the organizers of today's summit for the invitation to speak to all of you and share our research project. As the first talk in the animal health and welfare session, I think I'd like to begin by apologizing for my title, and with perhaps a particular apology to my chancellor. Nanosculpted silicon membranes for shape-based biological separations. It's a mouthful even for me, and it's my title. So in an effort to make it a little more approachable, I added a subtitle. Don't push square pegs through round holes. Now, admittedly, this subtitle doesn't evoke images of milking cows either, but I hope that by referring to this old adage, I'll be able to help you visualize how our nanosculpted membranes may be able to help dairy farmers. I'm going to begin by reviewing these round holes and highlighting the advantages that we believe our technology can provide. So in biology and chemistry laboratories, we can spend a lot of time trying to separate biological samples from one another. Traditionally, we do this by exploiting differences like mass and size. For example, if I have a mixture of complex materials, I can try to layer them on top of a solution in a test tube as shown on the left-hand side, and then spin that sample in a centrifuge. And samples that are heavier and denser will typically migrate further, and in this way you can see that I can separate them from one another. Another popular way to separate materials is to pass them through a filter. And filters are manufactured by creating different size round holes in a surface that allow smaller materials to pass through while inhibiting larger species. But here's the rub. Filters are characterized based on the average size of a material passing through the hole based on its mass. And so, the larger the hole, the more mass that it can accommodate. But this is somewhat restrictive. So consider this Wisconsin example. A butter burger from Culver's and a brat from State Street are, for the most part, the same weight. But if you ordered a filter based on that weight, your ability to pass those objects through the filter are going to be remarkably different. A smarter strategy may be to create pores that are based on the three-dimensional structure of the object you're trying to filter. And that precisely is the approach we're pursuing with our nanosculpted membranes. The real power of this technology comes from the fact that it is not confined to circular pores. As these images on the right show you, our pores can be squares or they can be long channels, and these samples are just a subset of the geometries that are achievable. And in this example, it seems pretty clear that the filters on the right are going to be preferable for separating brats from hamburgers. And I'd also point out that the geometry of these filters is on the nanoscale. And a nanometer is 1/1,000,000th of a millimeter. So what we're doing is combining the ability to make customized shapes with microscopic sizes to create a new type of diagnostic device. And the question here today is how this platform technology can be applied to the dairy industry. To the next slide, please. And so we're interested in a disease called mastitis. And mastitis is the inflammation of a cow's udder, and it's the most common disease in dairy cows. And being the most common disease means that it's also the most costly disease in dairy cows, costing farmers up to $200 per cow per year. And estimates based on lost production time run upwards of $140 million a year for the state of Wisconsin. Now, mastitis is challenging because there are almost 20 known different causes of the disease. And most of them come from different bacterial strains. And you're probably familiar with some of those. Streptococcus is a bacteria that can cause strep throats in humans. Staphylococcus is a source of staph infections. And E. coli is the darling strain of laboratory researchers and also the occasional culprit in food poisoning. And because there are so many varieties, farmers use broad spectrum antibiotics to treat infected cows. And in addition to the cost of the medication, this treatment regimen can result in the generation of an antibiotic-resistant strain of bacteria, which can be devastating for the farm. And so we're hopeful that our nanosculpted membranes can reduce that risk. The goal of our project is to create customized filters based on the specific geometries of these pathogens. And this will allow farmers to be able to apply milk samples directly to a cartridge-based filters that contains a stack of different shaped filters, allowing for the rapid identification of a particular pathogen and the immediate application of a targeted response. This, at the same time, should reduce the cost and increase the safety for our dairy cattle. And so with that, I'll thank you for your time and pass it on to our next speaker, my colleague, Carrie Kuhn from UW-Madison.
Thanks so much, Mark, and thanks to all of you for being here today. I couldn't be more excited to share a bit about the research that we are doing with support from the Dairy Innovation Hub in my lab at UW-Madison. And I'd like to start by acknowledging the people that are helping to make this work possible. So in particular, Garrett Soon and Joanna Elfenbein here at UW-Madison. And most of all, Andrew Summer, who is a PhD student in my lab and who is going to be responsible for actually conducting most of this work. So our research project broadly seeks to better understand the capacity of flies to acquire and transmit disease-causing bacteria to Wisconsin dairy cows. Now, for the last 10 years, and really the bulk of my career thus far, I have been working with a group of insects that are close relatives of flies, and that I'm certain everyone here is familiar with, mosquitoes, which of course are very important insects because several species transmit disease-causing organisms to humans and other mammals. And indeed, here at UW-Madison, my lab's research program has and continues to focus on understanding the function of bacteria and other microbes in regulating growth, reproduction, and immunity in mosquitoes. But more recently, my lab has developed an interest in expanding this work to include other insect disease vectors like flies. And so I suspect that those of you that regularly work or that have at least stepped foot in an active dairy barn are well aware that cows often share their home with a variety of biting and non-biting flies, which are a nuisance and a health concern for the cows because they may spread disease-causing organisms. So Maria, if you could advance the slide, that'd be great. So the flies themselves live in close association with potentially pathogenic bacteria. And this is because dairy cow manure is the primary site where flies breed and lay their eggs. So eggs laid by adult female flies hatch into tiny larvae, which feed on the surrounding manure and molt several times into larger stages. When the larvae finally reach maturity, they pupate and eventually emerge as adults. And so during this time, larvae also ingest bacteria present in the manure, which can persist to the adult stage. And these may include pathogenic bacteria like Staphylococcus, Streptococcus, Salmonella, and E. coli, which, as Mark mentioned earlier, are known to cause diseases like mastitis and enteritis in dairy cows. So again, as Mark mentioned, mastitis is characterized by severe inflammation of the mammary gland and udder tissues, while enteritis refers to inflammation of the intestine. And together, mastitis and enteritis are two of the most common and costly diseases to the dairy industry. They can cause a wide variety of problems for farmers. Milk from infected cows usually must be discarded, and infected cows must be medically treated. And this, results in pretty significant economic losses of up to $35 billion each year. So while a lot of attention has been focused on developing novel intervention and treatment strategies to manage the impact of these pathogens on cow health and production, relatively little is known about their epidemiology. So understanding how these pathogens persist in the environment and are transmitted to susceptible cows is an area really of high research priority. So Maria, you can hit the next slide. Thanks. So what our research will do is examine the presence and abundance of potentially pathogenic bacteria in fly and manure samples collected from dairy farms across southeastern Wisconsin. And then simultaneously with that, we will also perform functional assays in the lab to examine colonization and persistence of known clinical isolates in a fly model, which is the housefly Musca domestica. We're also planning to develop a series of informational materials related to our research to distribute to Wisconsin dairy farmers and stakeholders. So these materials will include physical pamphlets with general information about fly biology, the species of flies most commonly found in Wisconsin and how to identify them, and sort of a broad overview and summary of our research. Digital materials will also be developed as part of a mobile app to provide dairy producers access to an interactive key for fly identification, as well as interactive maps that display historical and current mastitis and enteritis incidence rates, and the distribution and abundance of bacteria of interest in fly and manure samples collected as part of our research. So collectively, our results will provide novel insights into the processes underlying environmental persistence and transmission of these bacterial pathogens, and will ultimately help farmers to raise healthier and happier cows in Wisconsin. So with that, I'll thank you again for your time and turn things over to Sylvia Kehoe from UW River Falls.
Hi, everyone. Thanks for the opportunity to speak about my research, and thanks for everyone that's attending and your interest in this area. So calf health and management is my research area, and I am very interested in doing research that not only helps raise the calf in a much healthier and friendlier way, but also to help producers, and make their life easier. When you're raising calves, the biggest thing about calf raising is they're a lot of work. Just like anybody that's had, babies and young children, they're kind of the same. And so as I was exploring some different topics with students, we came across the method of disbudding calves. And so disbudding, we used to use the word dehorning. Dehorning is not an accurate term anymore because most farmers will take the horn tissue off of a calf before 2 months of age. And after 2 months of age, that horn tissue has attached to the skull, and it's much more painful and difficult to do. And so farmers do that, relatively quickly after the calf is born. And there's two different ways, and so we call it disbudding. There's two different ways of doing this that's most common. One is the hot iron method, which is on the photo on the right-hand side towards the bottom. That's a hot iron where you put a lidocaine block, you give the calf a lidocaine block, and then you take the tissue off. And the other way is kind of a newer method. It's been around since the 1950s, but it hasn't really been used until recently. And it's called caustic paste. And so caustic paste is just a paste that has some hydroxides in it. So it's a really high pH. It's caustic. So it, kind of burns everything it touches. It's a chemical burn. And it is now currently one of the more popular methods of taking the horn tissue off of calves. And the reason for that is because it's a lot easier and quicker to do for the farmer than, taking out a hot iron and heating it up and doing the lidocaine block and everything. We also recommend a lidocaine block with the caustic paste method, but you don't have to deal with a big piece of equipment. And so when my students and I have a lot of undergrad students that want to do research, and when my students and I were looking into, this caustic paste method, we realized that there's very little research on the paste itself. So there's two main companies that produce caustic paste. They're shown on the slide. Dr. Naylor's is one that's been around for a long time, and then Dr. Larson's is another. And they have very general directions, which is, apply paste, in a quarter size to the calf bud or to the horn bud, and just apply enough, right? And so it's been quite the experience to realize that, there really isn't a lot of methodology work in this area. Plus, we also want to make this much more consistent application. And I'll show not yet, but in the next slide, the difference between good pasting and bad pasting. And we want to make it easy. So in my calf lab research, we've developed these little applicators. And so you can see the calf there in the photo, but they're just these 3D applicators that I'm working with John O'Baidlin from UW-Platteville with, and he is, we're collaborating on this where he's developed these 3D applicators for us. And we want to make this easy and consistent for the producer. We also want to make sure that the paste is covered And the reason for that is caustic paste, just as it sounds, is caustic to tissue. So it's supposed to do its job on the calf, but if the calf is housed with another calf, for example, and that's where the dairy industry is heading, is to house calves in pairs or groups, the calf could potentially rub its head on another calf and cause some issues on the skin of that calf. And also, because we do this, we want to do this as soon as possible to the calf so that it's, the younger it is, the less likely it is to feel as much pain and be able to move around and, rub it off. Then we are also worried about worker arms. So when you're trying to bottle feed a calf or you're getting it up to help it eat and give, different vitamin shots and things like that, we don't want this rubbing off on the arms of the workers. And so we've been going through these different applicator prototypes. We've been through about 5 of them now, and I think we're finally getting to a good applicator where we can apply it and make it easy and consistent. Next slide, please. And so, there's a number of things that we can do with this paste. And for one, we want to see, is there a paste that is maybe superior to the other one? And so potentially, are they both as efficient in taking the horn tissue off? Are they both, do they both cause the same pain behaviors? And they're both slightly different in their components. So we're looking at that. We're looking to see how much can we apply and still be successful. We assume that the less paste you can apply, the less painful it is for the calf. Even if we were to give a lidocaine block, and we typically give NSAIDs, like an ibuprofen, to help take the edge off of that pain, we try and make the calf as comfortable as possible. But obviously, if we can apply less volume, then that calf potentially will feel less pain, no matter what we're doing to help it. And then also, the age of the calf. So we know that we want to do it as early as possible, but when is that appropriate age? And then I'm working with a couple of people from UW-Madison currently, Dr. Van Ost and Dr. Adcock, where we are going to be investigating whether we can give the calf a pacifier to help reduce its pain. So we know that, babies and calves like to suck on things. And so maybe that would help reduce some of those pain behaviors for the calf. And, there's a lot of different research that we have here. And so you can see in that picture, the 2 pictures on the bottom, the calf on the left has a really large pasting site, and the calf on the right has a very small one. So again, we want to make this easy for producers. We want it to be consistent and, and make it as pain-free for the calf as possible. So thanks for listening, and I'll turn it over to Maria for any questions we might have.
Great. Thank you so much to the 3 of you. We do have a couple of questions to get to. I'll mention to our audience as well that you have a chat box in your media player, and that is where you should type your questions if you have them. So the first question is directed to Mark and Carrie, but it probably could apply to Sylvia as well in some respects. And that is, Mark and Carrie, both of you are not in dairy specific fields. So what drew you to research cows?
Mark, do you want to go first?
Sure, Carrie, I'd be happy. I am a biologist, a molecular biology professor here, and my grad training is in biochemistry. And I just really like to tackle problems. And so my collaborator and I here have developed a new technology that we believe is a platform that can be used in many areas of research. And when we learned of the Dairy Innovation Hub, we saw that there was a direct application that would allow us to advance this technology and be able to address issues that are important for our state. And so we made an appointment with Tara Montgomery, who you've heard about in the summit here today. And she encouraged us in that collaborative spirit to apply for a fellowship. And so it's really just, our curiosity to solve problems and to be able to apply them to this particular area.
Yeah, I love this question. So I'm also a basic biologist by training, but I've also been lucky to have always worked on systems, having worked on mosquitoes most of my career, that are always relevant and compelling for people in the community. But for me, I always sort of envisioned when I was building a research program in my own lab of always making sure to be doing things that were relevant to the immediate community at my university and the state in which my university is in. And so it was just sort of a logical, flies and their ability to transmit diseases is something that's come up time and time again at conferences and things in my own sort of little niche. And so it was sort of a logical jump to sort of take those ideas and apply them to a system in my home, my new home, right? That is close to home and that hits home and that is really important for people here. And I think also this has come up several times already that I really just want to say how grateful I am. UW as an institution is such a collaborative environment. And the Dairy Innovation Hub is doing nothing more than just to facilitate that. And so this has also been like a really great way for me as a newer PI at UW-Madison to collaborate, to form some really, really strong relationships, collaborative relationships with other researchers here that have been working in the dairy world for much longer than me. So I'm looking forward to this, to learning more about the dairy side of things and hopefully continuing to work in this long into the future.
And Sylvia, we actually got a question specific to your study, so I'll give that to you. So for the calf paste study, are you looking for farmers to try out some of your ideas? And then as a layup to that, they're also curious if the pacifier idea would lead to calves doing more sucking in a group setting.
Yep. So that is a common, I'll start with the second one first. That's a common misconception. And we always think about that calves are going to want to suck. And if we provide them with something, they're just going to really suck, and then they'll suck on everything else. But it's actually not true, where calves, they do have this need to suck on things. And so what we're doing in this case is we're providing them an appropriate outlet for that. So we're actually going to keep the pacifier in there for 3 weeks. And, we'll monitor if they do potentially, for some reason suck on other things, but that's been pretty consistently where the pacifier itself won't teach them that. There's other things, so a lack of nutrition, not enough milk, that kind of stuff, that's where those problems come from. But even with calves that don't have, they're potentially have that need to suck because they don't have enough nutrition, providing them with something appropriate to suck on would help with that, but it's not going to teach them to do it. So yeah, that was one of our farmers' concerns who's on the study, and we'll also we'll keep track of that, make sure that it's not happening. But as far as the applicator, so we've been through 5 different prototypes, and we're finally to the point where I think we're ready to test it. And but that that has to be done still by us around here. So if the farmer is potentially within driving distance, then we would be more than happy to have them try those out. But at this point, we're not quite yet to the point where we need farmers, but at some point we will be, and then I will be looking for help to test those out. So thank you.
Great. Well, thanks for those